Tonight's guest speaker, Dr. Heather Joesting, is an associate professor at Georgia Southern University's Armstrong campus in Savannah. Dr. Joesting holds a PhD from Wake Forest University, and currently she serves as the senior scientist for Georgia Southern's Sustainable Aquaponics Research Center. As a coastal plant ecologist, Dr. Joesting's research focuses on a variety of topics, most of them related to salt marsh and coastal sand dune plant species. Tonight, Dr. Joesting will be telling us about her efforts to cultivate and propagate smooth cordgrass for salt marsh restoration projects. And this ties in really well to a lot of the work that we are doing at Florida Oceanographic Society. So it is with great, great pleasure that I introduce all of you to Dr. Heather Joesting.
Okay, thank you so much for that introduction. And it is my pleasure today to be here to tell you guys about some of this exciting research that my lab has been working on to basically produce these Spartina plants for salt marsh restoration. So as many of you know, coastal salt marshes are extremely productive habitats, and they are found fringing the eastern and Gulf Coast of the US. In the continental US, there is a combined 1.7 mega hectares of salt marshes. And as you can see from this graphic, the majority of these salt marshes occur along the east and the Gulf Coast of the US. And you can even see here, especially in the southeast, including Georgia and Florida, we have extremely extensive salt marsh communities. Just to give you some numbers, along our 100-mile coast of Georgia, we have 400,000 acres of salt marsh. And for you guys in Florida, you have a coastline that spreads about 1,350 miles, and you guys have 1.3 million acres of salt marsh. Salt marshes are also very significant habitats in that they provide numerous ecosystem services. They support the high diversity and abundance of other organisms by serving as raw material for food, basically through that rich expanse of salt marsh grasses. They can function to protect the coast from storm and wave energy by acting almost as like a buffer that dissipates that energy before it reaches the shoreline. They can function in carbon sequestration and are important in maintaining commercial fisheries by acting as a breeding ground and a nursery ground for juvenile fish species, particularly those that are of commercial importance. And finally, they not only export nutrients to the marine habitats, but they actually filter that water before it reaches those marine habitats and takes up pollutants and excess nutrients that could cause harm to marine resources. Uh-oh, sorry, mouse stopped working. Okay, so along the East Coast and the Gulf Coast, the smooth cordgrass, or as I'm going to refer to it, Spartina, alterniflora, is the dominant salt marsh species that we find in these marshes along these coasts. Spartina is very well adapted to exist in what we consider a pretty stressful habitat for plants. This plant has mechanisms that allow it to tolerate soil salinity, as well as tolerate the low oxygen availability in the soil. Some of the cool things that I think that this plant can do is it has actually created these big open air spaces in its stem. This helps it to diffuse oxygen throughout the plant. And it also is able to sequester salt and then secrete it out of glands on the leaves. And so oftentimes it hasn't rained for a little while, you can walk by these grasses and just pull your hand up along the blade and pull out salt crystals. So these plants are very dominant. Oftentimes when you look out at the salt marsh and you see that sea of green, a lot of that green is the Spartina. And it plays a very important role in the ecosystem. And one of its most vital roles is its role in facilitating horizontal and vertical growth in the marsh. This is how the marsh grows and maintains elevation and expanse. If you look at this kind of image here of this Spartina plant, you can see these underground structures. These are underground stems called rhizomes. And these rhizomes will help stabilize sediment to keep it from being washed away. This contributes to the horizontal growth of that marsh. Additionally, the upright growth of Spartina facilitates sediment deposition by acting as a barrier. This will help to increase the vertical growth of the marsh. Spartina is also a significant contribution to the high primary productivity of the salt marsh. Its contribution to primary productivity just increases as you go south and the growing season becomes longer. And it also supports, as I mentioned, all the other organisms that we find in the marsh ecosystems by forming the base of the green and brown food web. So the green food web is your typical consumer driven plant consumption food web. But we also have a very significant detritus driven food web where there are a myriad of organisms that subsist off of decomposing dead Spartina stems. Unfortunately, there's been a global decline in salt marshes, and this has been due to a myriad of factors. Some of these include reclamation of marshes for human development. So these marshes have been filled and turned into some type of development. There has been some dieback events along the Gulf Coast and the East Coast of the US, where these large, you can see in this image here, these large swatches of marsh grass will just turn brown and die. And once those plants die, then that soil becomes more prone to erosion. This has happened, at least here in the Georgia coast, several times in the past couple decades. I couldn't find any recorded events there in Florida, but it has also occurred in Louisiana. We have habitat degradation of these salt marshes due to pollutants and altered hydrology as humans alter the size or the location of waterways to suit shipping needs. And this causes changes to the water flow into these marshes. As with many ecosystems, there is a threat of biological invasions that can change the structure of the system or actually change the trophic and feeding relationships. And then, of course, one of the biggest threats to salt marshes currently is inundation to sea level rise. And so you can see here along this map that our areas of Georgia and Florida are kind of, especially on the East Coast, we are under very high or high vulnerability to sea level rise. And sea level could cause the complete destruction and inundation of these systems. So in order to avoid losing salt marshes to sea level rise, then we have to see these marshes grow vertically and horizontally at a rate that is faster than that of sea level. So in this, to try to preserve these systems in the face of sea level rise and to preserve these ecosystem functions, there has been recent efforts to restore these habitats. And living shorelines is a restoration technique that is being promoted here along the Georgia coastline that combines green infrastructure and native biota to stabilize shorelines. And you can see this is an example from the Georgia DNR this is a project that has been done at the Ashantilly. And you can see how this restored marsh, living shoreline is kind of restored that natural habitat. There are a lot of advantages to living shorelines. First off, everything is natural and it's going to mimic natural shoreline dynamics. It also creates that buffer of vegetation that reduces erosion and stabilizes those shorelines. And it returns some of those ecosystem services, such as providing and maintaining habitat for aquatic plants and animals, as well as restoring that critical feeding and nursery habitat. Along our coastline, we often include Spartina as a biotic component in these living shorelines due to its role in vertical and horizontal accretion of the marsh, as well as the maintenance of marsh ecosystem functions. So Spartina has a really, really big geographic distribution. And so as you can see from this map, it is found from New England all the way down to the Texas coast. And so there has been some studies in looking at this distribution along this latitudinal gradient, because we see a lot of abiotic differences in environmental variables along this gradient. From this, researchers have discovered that, at least looking into the genetic relationships among different populations along this complete range, they've identified 4 distinct groups of Spartina. They have discovered a New England group, a Mid-Atlantic group, a South Atlantic group, and a Gulf Coast group. And all of these are genetically unique from each other. You can see that Georgia and Florida over here are in that South Atlantic group. These groups have been shown to have variations in phenotype. This includes morphological variations like the size of the plant and the density of stems, physiological variations, as well as reproductive variations. In the study by Seliskar et al. In 2002, they collected plants from marshes in Massachusetts, Delaware, and Georgia, and they planted them in a common garden in Delaware and tracked kind of their growth and productivity over time. What they found, if you look over here, where my pointer is that even though they were all kind of planted in the same similar garden, each of these, what we call ecotypes, maintained their phenotypes that were found in their native range. For example, in the native range, Georgia Spartina plants have been found to have taller stems than Massachusetts and Delaware. And in that common garden experiment, this phenotypic difference was maintained. We also see that Georgia has taller plants, but they have less density of stems. And again, this was found in natural marshes and again, replicated and shown again in this common garden experiment. What this means is that these phenotypic differences are not necessarily just environmentally controlled, but are actually genetic unique differences. So what we would think is that because the ecosystem functions of this plant are so influential, that having a phenotype that is different from what is normally placed in that marsh may cause problems with ecosystem function. For example, if I planted a Massachusetts plant in a Georgia marsh, it may have those phenotypic differences that don't support that community the way a Georgia plant would. Based on this, recent studies have suggested that the locally sourced plants will actually increase your restoration success. Some studies suggest that those plants should be sourced at no more than 100 kilometers away from the restoration site. In addition, genetic variation has been linked to some of these plant functional traits that affect how erodible the soil is. And so it's been suggested that high genetic variation will increase restoration success. So when we look at the genetic variation within salt marsh populations in Georgia, we see that our populations have very high genetic diversity. So traditional nursery production methods for Spartina often include what we call clonal propagation. And so how this happens is you have this underground stem that I showed you before. This is the rhizome. And along this rhizome, these independent stems will be produced. So here you have 1, 2, 3 stems, and these can continue to be produced along this underground rhizome. Each of these stems, because they are connected by this rhizome, are genetically identical to each other. Oftentimes, to produce a nursery stock, these rhizomes are collected and cloned in order to produce a nursery stock. It is a very easy way to propagate this plant, but unfortunately, because you have a lot of clones of the same genetic individual, this can lead to pretty low genetic variation within your nursery stock. However, if these were propagated by seeds, which are produced through sexual reproduction, this would create genetically unique individuals that would be part of your nursery stock. And that would, again, enhance the genetic variation within that nursery stock and possibly increase the success of that restoration. So really, the purpose of our research here is to develop seed propagation protocols for Spartina. We hope that these seed propagation protocols can be used by local plant nurseries to produce Spartina plants for restoration in Georgia. So along these lines, I did want to mention just a couple of our experiments that we've been working on so far. So we've been working with Spartina and propagation from seed now since 2019. So the kind of 3 experiments I want to talk to you about today are some of the ones that we are either completed or are working on. So these include characterizing the fecundity, which is the reproductive output, and the phenology, which is kind of the flowering times of Spartina in coastal Georgia, because we'll see that there are these phenotypic differences also include differences in reproduction. Second, we looked at producing Spartina using above ground wetland to remediate aquaculture effluent. So we looked at some innovative techniques to produce Spartina. And finally, we are currently working on determining best practices for seed propagation of Spartina. This research is being funded by a Georgia DNR Coastal Incentive Grant and being done in collaboration with Joel Koska at Georgia Tech. So first, let's start with our first kind of study, which is our ongoing attempt to characterize fecundity and phenology of Spartina. So as I mentioned previously, we have these phenotypic differences in these groups along the Atlantic and Gulf Coast of Spartina. And those phenological differences also include differences in reproduction. Specifically, we see phenological differences, which means that their time they flower during the year, as well as the length of their flowering season, varies along that latitudinal gradient. And we also see differences in energy allocation. So this is where those resources are being allocated. Is it being allocated to reproduction? Is it being allocated to growth? Is it being allocated to storage? And it has been shown that the further north you go, the more of energy that's being allocated into carbohydrate reserves to make it through that long, cold winter. So if you had more investment into these storage reserves, that would leave less for reproduction. We may also see variation in reproduction within a single coastal region. For example, you may see variations in salinity that has to do with proximity to the ocean that may result in variations in reproduction. And of course, we may expect to see year-to-year variations due to climatic variables. And that was one of the things that we were really interested in with this study, is just to kind of characterize what these spatial patterns and reproductive output may be, as well as these annual patterns. And this supported the nursery production, and that we were hoping to kind of give a guideline on when is the best time to collect these seeds. And so that is part of our purpose, was to determine the best time of year and the best location within a coastal system to collect seeds, and to characterize those patterns in coastal Georgia. So to do this, we selected 3 marshes with varying marine influence in Chatham County, Georgia. So just to kind of get you situated, Savannah is actually up here. And so our closest site to the marine influence was here at Lazaretto Creek Boat Ramp. This is near Tybee Island, Georgia. We had another site that was further into the Marsh, which is this Rodney Hall boat ramp here. And then our site that had the least marine influence was a marsh in the Halcyon Bluff Community House. This is a neighborhood marsh. In 2019 and 2020, we visited the marshes every 2 weeks between October 1st and November 31st. While we were at those marshes, we collected seeds if we could find them. And we also assessed reproductive status for plants along a transect to kind of get an idea of flowering times and phenology. Just to give you a better look at how these marshes differed. So first we had that Lazaretto Creek boat ramp that is shown right here. Lazaretto Creek boat ramp is right before the bridge over. So you can kind of see this bridge back here. Once you go over that bridge, you're on Tybee Island. We found salinity varied very little and stayed between 25 and 26. And this site is a public use boat ramp. It has double lanes here. And you can see that we have a fringe of the tall form Spartina. And then we have this huge expanse of short form Spartina behind it. So we collected seeds in this expanse back here. We could not make it to the fringe without sinking very, very deeply in mud. Our second site, the mid affluence, is this Rodney Hall boat ramp. The salinity here varied between 15 and 24. As you can see, this is also a public boat ramp, except it is much larger than Lazaretto. This actually has 4 lanes, so each of these 2 lanes are double lanes. This site gets pretty extensive boat traffic and is really frequently used. And it's inundated right now, but we collected in this fringe of tall form Spartina that occurs here along the fringe of this boat ramp. And finally, our last site is not as good as a picture because I had to take an image of this using Google Earth, and we probably had that taken during the winter. But this is our site with the least marine influence. This site is actually in my neighborhood, so I made it kind of easy to get to. But we have a community house right here for the neighborhood, and so we collected kind of in these marshes behind that community house. This site was also dominated by that taller form Spartina. So at each marsh site, we collected 20 to 30 mature spikelets when they were available. A mature spikelet is this structure right here. So it's basically just the stem of a Spartina that is reproductive. And at this time, the spikelet is mature because it has mature seeds, so it's ready to be collected. You can see the seeds right here. So once we collected those seeds, kind of stuck them in a plastic bag until we could get back to the lab. And then we removed the spikelet by basically just chaffing them very similar to how you do with wheat. And then counted the number of seeds per spikelet. Once we counted those seeds, we placed them in cold, wet conditions. So we put them in water and placed them in a refrigerator at 4 degrees Celsius, where they remain for at least 3 months. Spartina requires at least 2 to 3 months of this, what we call cold wet stratification, in order for the seed to break dormancy and be ready to germinate. After that cold wet stratification, we tested the viability of the seeds using what's called a tetrazolium test. The tetrazolium test is basically just the use of a dye, and that dye will stain metabolic activity. And so if we had a growing embryo in these seeds, then its metabolic activity would get stained. So if you look at this image here, these are 2 Spartina seeds. This is before soaking in the TZ solution, and this is after 24 to 48 hours. You can see this red coloration here is indicative that there is an embryo in the seed and it is viable. The only downside with the TZ test is it does kill the seeds. And so we tested this on 25% of the seeds from each of our spikelets. If those spikelets had seed viability greater than 10%, we then put them under germination. We did this by placing the seeds in culture dishes with water, where they remained in water and were monitored for 14 days for germination. Once those seedlings had germinated and they were about 1/2 inch tall, we would move them to peat pellets, and they would stay in these peat pellets in a sub-irrigated tray with a humidity dome to promote seedling growth. And after we maintain those in lab conditions for a couple of weeks, we would then move them out to the greenhouse and monitor them for future survival and growth. For our phenological studies, we did this only at a single site in 2020. And to do this, we took a transect and we established a transect parallel to the shoreline on the front part here along the creek bank and along the back edge of this marsh. Along each of these transects, we placed 0.5 meter square quadrant, which you can see my student doing here, every 2 meters along this transect. And within each of those 2 meter quadrats, we counted the total number of stems, and then we counted the total number of stems that were in one of the following reproductive statuses. They were flowering, seeding, or fruiting. So if we look at kind of the results of our reproductive output, we did find that the location of the marsh and how close it was to that ocean influence did have a significant effect on not only how many seeds were found per spikelet, but also their viability. So if you look at this graph, you have these lines with these different letters. Sites that are grouped together are not different from each other, and letters that are different are significantly different from each other. So what we see with this first graph is we can see that Halcyon Bluff Community House, which was the site that was furthest from the ocean, had significantly greater number of seeds per spikelet than the other two sites. When you look at seed viability, we can see that there was significantly greater percentage of seeds viable at Halcyon Bluff Community House and the Lazaro Keep boat ramp. And we had significantly lower viability at that site that was kind of that intermediate in marine influence. We also have found a significant effect of year on our number of seeds per spikelet. And so again, you can see here that in 2019, we had greater overall seed numbers compared to 2020. And we also saw a significantly greater germination rate in 2020. So we saw a germination rate around 10% in 2019, and we got down to about 3% in 2020. So these germination rates sound extremely low, and they are. This is one of the reasons why we're trying to optimize these germination conditions so that we can bring these numbers up. However, Spartina is kind of known to have these really low germination rates. And so getting a rate of 10% is not uncommon. Along the Gulf Coast, oftentimes germination rates there also around 10 to 15%. And finally, we can see that in 2020, we had a significantly greater survival rate of our seedlings. We saw almost a 70% survival rate compared to 20% in 2019. If we look at our phenology results for 2020, we can see that peak reproduction really started to occur in October. So the these black bars here indicate the percentage of plants that were flowering on every given date. And you can see that we peak our flowering around early October. The gray here shows the development of fruits. That would be kind of this intermediate between flowers and seeds. And you can see that that kind of goes on year round, but we get an increase in the percentage of plants that have seeds that are ready for collection in mid to late October. So what can we conclude from this? Well, interestingly, we did find that there are spatial patterns in fecundity or reproductive output. Again, we saw the greatest number of seeds per spikelet and the greatest seed viability at the marsh that was furthest from the ocean. We believe this has something to do with the reduced stress that exists with the lower salinities. So this kind of image over here guys kind of shows a plant's energy allocation. Okay, so what isn't used by a plant in just basic maintenance of plant activities is considered surplus energy. And that surplus energy can be allocated to growing and getting larger, or it can be allocated to investment and reproduction. And that investment and reproduction would include increased flowers and increased seeds and increased size of these flowering and fruiting stems. But when we get to the salt marsh, we have an added allocation. So in this area with high abiotic stresses, like soil salinity and low oxygen in the soils, that means some of this energy has to be allocated to dealing and physiologically responding to those stress factors. So any energy that's going into this physiological response for survival is no longer available for either of these other buckets. In sites closer to the ocean, where salinity is perhaps is higher and closer to that of saltwater, we can see that there's probably more energy allocation going to dealing with this physical response and less in the reproductive investment. As we get further away from that ocean, we have less salinity. And so the plant is able to take some of that energy and allocate it into reproduction, creating just these larger flowering inflorescences that result in a larger number of seeds per spikelet. One of the other interesting things that our study showed was that our intermediate site not only had very low number of seeds per spikelet, but it also had the lowest, significantly lowest seed viability. We kind of think this might have something to do with anthropogenic disturbance. And so if you remember those different sites, 2 of them were boat ramps, but the Rodney Hall boat ramp, which is our intermediate site, is very heavily used. There's a lot of boat traffic, and especially on beautiful weekend days. And research has shown that boat wake can have a significant impact on the shoreline. Oftentimes marshes are found along low energy coastlines. And when you have frequent boat wake, it can cause that energy to kind of exceed that threshold for marshes and can cause increased erosion. So this increased boat wake may be just causing enough disturbance in this site that these plants are not able to put so much energy into reproduction. And again, boat wake has been shown to contribute significantly to shoreline erosion. And this occurs where boat activity is regular, concentrated, and close to the shore. And all of these are occurring along that fringe marsh there at Rodney Hall. So based on kind of the spatial patterns, we would recommend that seeds with the lowest marine influence or furthest away from the ocean may be the best place to collect seeds, because you would increase your number of seeds collected as well as the potential viability of those seeds. We also found annual patterns in fecundity. So if you remember, we found that there were more seeds per spikelet in 2019 overall than in 2020. We think this might be linked to climatic variables and the pattern of rainfall between those 2 years. As you can see, 2019, although we have very little difference in the total precipitation between 2019 and 2020, we do have a big difference in kind of the pattern of that precipitation. June was a particularly wet month in 2019. So we had a lot of fresh water reserves here with a lot of rainfall. What the rainfall can do is it can not only rinse the salts off of the Spartina stems, but it also can dilute that seawater and cause that salinity to decrease a little bit. And we see that in 2019, not only was it a wetter summer, but we also had some increased rain as well during that flowering season. So it could be that just this pattern of rainfall, this large amount of rainfall between June and November, may have contributed to more reproductive output. We also saw greater germination rates in 2019. This could be due to those climatic variables as well, but we think this might actually been a result of fungal colonization that destroyed about 30% of our stored seeds. We had decided to try to keep the lids unscrewed a little bit to allow oxygen to get into the seeds. But unfortunately, that oxygen caused fungal growth, and we ended up having to toss about 30% of those seeds. Those that remain may have still had some of that fungal growth in there, but we just didn't visualize it. And finally, we saw greater seedling survival in 2020 compared to 2019. So these years that I'm using are their collection seasons. And so we were actually starting to germinate our 2019 collection in the spring of 2020. And this is right when the pandemic hit and things went under quarantine. And so I feel that probably the lack of attention that our seedlings got during that spring 2020 may have led to that overall lower survival. If we look at our phenological data, we see that at that maximum reproduction, we actually only had 25% of our plants on average that were reproductive. And this was done by kind of looking at each of those quadrats. and looking at the percentage reproduction within each of those quadrats and then kind of averaging them all together. So we see that there at one time during this 2-month period, the greatest number we had was about 1/4 of our population were in some reproductive states. This graphic here just kind of takes some of that information from the graph and just kind of shows where these peak time periods are for these different reproductive states. We can see that peak flowering occurred here in early October, while the peak time for mature seeds was here in mid to late October. So in order for recommendations, again, if you kind of only had a few trips as a nursery to go out and collect your seeds, we would suggest going to sites further away from the marine influence and collecting seeds later in October. However, there is a caveat. This is based on 1-year data. And so we certainly need to continue monitoring the phenology over the next couple of years in order to kind of make up for those year-to-year variations that we might see. All right. So our second study was provided the second chapter for one of my graduate students' thesis. And this was looking at kind of these innovative ways to produce Spartina. And so we wanted to look to see if we could use an above ground wetland that was designed to remediate aquaculture effluent to grow Spartina for restoration. This was partially funded by an internal grant to my graduate student from the College of Science and Mathematics here at Georgia Southern. So we actually tied this into aquaculture. So as Zach mentioned, I am also the senior scientist for our Sustainable Aquaponics Resource Center here at Georgia Southern. And we've been looking at kind of innovative ways to increase the sustainability of aquaponics. And it kind of gave me this idea that instead of growing vegetables using this aquaculture effluent or wastewater, that perhaps we could also use it to grow plants for restoration. Aquaculture, if you aren't familiar with it, is basically fish farming. So it's the production of fish in these closed systems. And so this is just an example of an indoor system where there are fish being produced in each of these tanks. Aquaculture is lauded because it does reduce that fishing pressure that we see on wild fish stocks, but it does face quite a bit economic and ecological challenges in order for it to be developed into a really competitive industry. Some of those challenges have to do with cost. So what you see here is actually what's called a recirculating aquaculture system. So the water gets circulated as often as it can and cleans, but in tanks this size, as we know, fish tanks are going to periodically and regularly need to have water changes to keep the water quality optimal for fish growth. Oftentimes, when we're talking about water changes in these aquaculture facilities, we're talking about water change, even 50% water changes in 250 to 800 gallon tanks. And so that is a significant amount of water being used. In addition, when this water is removed during these water changes, it is very high in nutrients and has to be disposed of in a manner that will keep it from causing a pollution. So if these high nutrient wastewater from these fish water changes goes out into native aquatic ecosystems, it can cause eutrophication and habitat degradation. So many aquaculture facilities have an extra cost of proper disposal of this waste effluent. And finally, if we're talking about marine aquaculture, then there is a cost of salts as well, because every time you do a water change, you have to add more salts to get that salinity again back to that optimal range for fish growth. Now, similar to how vegetables can be used in aquaponics to remediate water, constructed wetlands have been shown to also efficiently remediate aquaculture effluent. Some of these will just dump that effluent directly into a wetland and allow it to be remediated that way. Or other ones will actually circulate it with the fish tank, similar to an aquaponic system. So we would have the water from the fish tank circulating through a wetland and then returning back to that fish tank in a closed loop system. Spartina is a perfect species for these constructed wetlands for several reasons. One, it is a native wetland plant, so it is accustomed to growing in those kind of wetland conditions. Second, Spartina can grow in both freshwater and saltwater. And so you can use these constructed wetlands not only to remediate freshwater effluent from freshwater aquaculture, but it can also be used to remediate marine aquaculture wastewater. And finally, like I mentioned, these plants are accustomed to being in saturated soils and can tolerate the subsequent low soil oxygen that occurs due to that constant saturation. So it seems like this plant would be a good fit as a choice for these constructed wetlands to remediate aquaculture effluent. From the nursery production side, traditional nursery production of Spartina includes growing them in pots in sub-irrigated trays. And so this is an image from the University of Southern Mississippi Center for Plant Restoration, where they grow several species of marsh grasses for restoration purposes. And you can see that these plants are placed in pots in these sub-irrigated trays. And so these trays need to have water maintained in them on a daily basis. So you can either put a drip irrigation system up, or someone has to come in and check the water levels every day. And they also need to be fertilized monthly using a commercial fertilizer. So on the nursery production side, producing these plants in constructed wetlands will reduce costs associated with their production. It will reduce the labor cost of making sure that those trays stay sub-irrigated, and it would also provide free fertilizer from the fish poop. In addition, that growing condition may be more similar to a natural marsh and may actually give them a little bit of an extra push in preparation for transplant into a salt marsh, especially if you use a constructed wetland connected to a marine aquaculture system. You would already have them salt hardened where they can be easily transitioned into the marsh. So the purpose of this experiment for us was to basically figure out if we could grow Spartina in a constructed wetland. Our wetland was designed at this time to remediate freshwater aquaculture effluent, just because we have not yet built a greenhouse cover over our wetlands. So what we would do is we compare the growth and productivity of Spartina in the wetland to plants that were grown using traditional methods. And as kind of a proof of concept, we also wanted to determine the efficacy of the Spartina itself to remediate that aquaculture effluent. So this is our basic design. So as you can see, we couldn't currently use a saltwater system because we do not have a greenhouse cover over this marsh, which means that the salinity would fluctuate too greatly with rainfall events. So for this research, this wetland was attached to a freshwater recirculating system, and that system was stocked with juvenile tilapia, which is the fish species that we use in our aquaponics facility that's located in this greenhouse over here. You can see this is a diagram of our constructed wetlands. So the wetland had 4 independent 4x8 cells, so we had the aquaculture effluent that would cycle out of the tank into each of these cells, and then it would cycle back out of these cells and back into the fish tank. Within these cells, we stocked 2 of those cells with Spartina, as you can see down here, and we left 2 cells non-vegetated. This was in our attempt to look at the efficacy of the water remediation potential of Spartina. To look at that water quality, we did collect water samples weekly, and every week we would analyze them for various water quality parameters. This included things like ammonium, nitrate, phosphorus. We looked at pH to make sure that was maintained. And we collected that from each of the cells as well as the fish tank itself. For the plant productivity, in addition to our plants that were located in our constructed wetlands, we also had plants that were being grown using those traditional methods of sub-irrigation and monthly fertilizer on 2 4x8 foot tables that were erected right near the wetland. Each week, all these plants were measured For plant growth, this included things like stem height, number of stems, and stem diameter, as well as productivity, which was, we measure chlorophyll content in the leaves, which is kind of a proxy for photosynthetic potential. I should state here that we did have to kind of boost up our stock of plants by collecting rhizomes. So each of these, we didn't have enough seedlings to completely stock and perform these experiments. So we went and collected some rhizomes and cloned and vegetated some from rhizomes and made sure that in each of our cells, we had an even mixture among all of our treatments of rhizomes and seedlings. So each cell in the wetlands and each plant table got the same mixture of seeds and rhizomes. So my students did this for 12 weeks in the grueling hot summer of Georgia. And at the end of 12 weeks, all these plants were harvested. They were cleaned and sorted into below ground and above ground, and then they were dried and weighed for biomass. So we had a lot of variables here, and when we went to analyze them, we wanted to kind of take each of these plants and look at how, with all of the variables that we measured, we wanted to look at how they kind of distributed themselves in a three-dimensional space. So we ran what's called a principal component analysis, which does just that. So I'm going to talk you guys through this. So what you see here is each of these symbols represents an individual plant. Those plants that are, those symbols that are filled are our wetland treatment plants in both red and blue. And those that are unfilled are our traditional plants. The blue colored symbols are seedlings, and the red color symbols are rhizomes. So once we kind of took this and we scattered these in this three-dimensional space based on these variables that we measured, we then looked to see if we saw any patterns of separation in any of our variables. And so the two variables we got interested in was, of course, our first objective, which was to see if there was a difference between the wetlands and the traditional methods. As you can kind of see in this image, we have pretty much the wetland and the traditional method plants all kind of scattered together. So there's really no separation between open symbols and closed symbols. They're all kind of mashed together. This suggests that there's really not a difference in the plants between those that were grown in traditional methods and those that were grown in the wetlands. However, we did see a clear separation based on the source. So if that plant came from a seedling or if that plant was a rhizome propagated plant. And so we could see that we had this kind of separation occurring based on the source of the plant. With the principal components axis, analysis, we can then see which variable or combination of variables are responsible for the pattern that we see in our data. We had two principal components that really explained most of the variation in our data. The first one was this first principal component. This is going to explain the spread of our data along this x-axis. And this component was comprised of a combination of three of our measured variables: growth rate, stem diameter, and leaf chlorophyll content. We also then saw that our second principal component, which explained the spread along the y-axis, was a combination of the percent change of stems and the root shoot ratio. So what this basically means is that differences in the combination of these variables on both these component axes are causing the separation between our seedlings and our rhizomes. So we wanted to explore this a little more. And so we ran statistical tests on these results. What we found is, again, there was no significant difference in growth and productivity of plants grown in the traditional methods versus the wetland. So this suggests that, that the plants in the wetland grew just as well as the plants using traditional methods. We did, however, see those significant differences between the performance of rhizomes versus seedlings. In all of our measured, and most of our measured variables, we see that there were significantly greater growth and productivity in our rhizomes and in our seedlings. You can see there's significantly greater growth rate, percent change in stems, so this would be stem growth over time, stem diameter, and chlorophyll concentration. What this suggests, it's not really a surprising result because the rhizome plants were more established plants, probably had more storage reserves, and could kind of tap into those to grow faster, while the seedlings were kind of starting from the beginning. What this really suggests to us is that we just need to give those seedlings a little more time to reach these points where they would be ready to be transplanted into an actual marsh. Now, just again to support our proof of concepts, we did find that all of our water quality parameters were kept within range of what we consider a healthy aquaponic system, but we did not see any differences in water quality parameters between our vegetated and non-vegetated cells. This was also not particularly surprising. We didn't really have complete isolation of these cells since the space and funding permit just a single tank of fish attached to each of these wetlands. And also each of these wetlands, there's one thing you can't stop, and that's algae growth. And so even the unvegetated cells had an abundance of algae that could contribute to remediation as well. So our conclusions from this is that you can use these constructed wetlands to successfully produce nursery stock of Spartina plants. And the Spartina plants can be used to effectively remediate aquaculture effluent. So this can be a workable system and an alternative for aquaculture systems to that, so they don't have to do that disposal. A kind of unexpected other result we found was in the creation of this wetland, we basically created like this little wildlife habitat. We ended up finding a vast diversity of organisms that were kind of hanging out around our wetland that we hadn't seen before. So this is kind of, all these dots here are frog eggs. We created basically a frog haven. They loved it there. We saw lots of grasshoppers and spiders and other insects, dragonflies began to kind of hang out around our wetlands. We started getting these huge black slugs. These are velvet leatherleaf slugs. I had never seen these before, but they were very common there. And this was the sound of coming to the marsh in the morning. So we would just have these chorus of frogs. And so it was a really kind of nice little habitat we had set up. So the next steps of this is to kind of continue the use of this wetland to look at nursery production. This summer, we are going to look at the effect of inoculation with marsh soil on Spartina growth. So our idea is to have half of our plants just grown in a 2-to-1 topsoil sand mixture, and the other half in that same kind of potting mix, but we're going to inoculate them with marsh soil to see if the microbes associated with that marsh soil can actually improve the growth of our plants. In 2023, we will finally be able to use our DNR grant to get a greenhouse cover over this wetland. And so we are going to actually simulate natural marsh conditions by using a recirculating system with a marine finfish. And we're going to actually try to simulate Georgia marsh conditions and then just look at how the growth and productivity compares from our locally produced plants versus those from various other sources. So finally, the project that we're currently working on right now is this funded project by the Georgia Department of Natural Resources. And it is to look at the best practices for seed propagation for Spartina alterniflora. My collaborator, Joel Casca, is working more on looking at genetic variation within not only native marsh populations, but also reaching out to other nurseries around the region. Most of these are in South Carolina and Florida to kind of assess their genetic diversity within their stocks. And my job is to collect seeds and try to figure out the best conditions for their storage and germination. So we've kind of repeated some of those seed collection methods that I showed you previously. We've just added yet another year to our collections. We added a fourth marsh location, which is kind of right here in the middle between our intermediate and our closer marine influence. This is called Priest Landing, and we are in this kind of short form marsh along here. This is this pier belongs to Skidaway Oceanographic Institute, and so this is more of a research pier than it is a public use pier. We also decided to test our collection methods by looking at how the vessel that you store the seeds in affects viability. Our idea here is that we're thinking about how easily oxygen can diffuse through these various storage vessels. So we stored our seeds in 3 different types of vessels. We use these plastic tubes, we use glass vials, and then we use plastic bags. And so we store them in those 3 conditions and to see how that would affect their viability. We are currently testing seeds for viability. We've been doing this since about mid-January. We collected a total of 40,000 seeds in fall of 2021. So it's taken us a little bit of time to get through all those viability tests. And we just started germination trials about a week or 2 ago. And what we're doing right now is we're looking at difference in temperatures. So we have some seeds germinating at a low temperature of about 72 to 75. We have others germinating at a constant temperature of about 85 to 90. And then another one where we have fluctuating daytime and nighttime temperatures to see how that affects germination. We do have some preliminary results from this year's collection, where we do see that when it comes to the number of seeds per spikelet, once again, we see significantly greater number of seeds in that marsh site with the lowest marine influence. So again, we're seeing that Halcyon Bluff is significantly greater number of seeds per spikelet than any of those other sites, almost double the amount. Our current results in seed viability, like I said, we're only part way through those 40,000 seeds. We again see that there is a effect of site on our seed viability. And again, as we have seen before, Rodney Hall Boat Ramp is just significantly lower seed viability than any of the other marshes. Even one that had kind of the Priest Landing area kind of had a similar salinity, but wasn't quite as disturbed with boat traffic as this Rodney Hall. We also surprisingly found a difference in viability in how we store the seeds. So we found that seeds that were stored in Ziploc bags has significantly higher viability than those that were in centrifuge tubes or scintillation vials. And so this is kind of interesting. We kind of hope this trend continues because this is, something that we may not think of, that it makes a difference what you're storing them in, but it appears it might. One of the other things that I've got an undergraduate working on in my lab is to look at how to maintain seedlings once they have sprouted. The traditional method here that we've been using is we place the seedlings once they get about a ½ inch tall in these peat pellets. We then sub-irrigate this tray so these peat pellets stay in water, and then we put a humidity dome on them to maintain humidity. Recently, Fork Farms gave our lab a flex farm hydroponic system on a long-term loan for research. And so we're interested in seeing how survival rates compare between these traditional methods and maintaining seedlings in this hydroponic system. So that's something that we're just getting started. We just started germinating seeds for this last week and are excited to hopefully stock this thing with Spartina seedlings here in the next 2 weeks or so. So with that, I have lots of people to thank. I want to thank my collaborator, Joel Koska at Georgia Tech, I also want to thank the University of Southern Mississippi Center for Plant Restoration. I did my postdoc with Pat Biber, who is the PI of the center, and their propagation guides for various coastal plants has kind of been a foundation for us to start looking at our own propagation protocols. The research that you saw was thesis research for 2 graduate students, Alexander Gregory, who is currently finishing up his thesis. He did the work on the nursery production in the wetlands, and Justin Henson, who is currently helping to test viability on 40,000 seeds. We also have a team of undergraduate students that have assisted in field collection, seed processing, seed viability testing, fixing up the wetland, and conducting that summer research. I also want to point out one of my new favorite little research assistants right here. This is my daughter. I've started bringing her out to the field to help us with seed collection. She's super excited. And of course, I want to thank my funding sources, the Georgia DNR Coastal Incentive Grant, as well as the Georgia Southern University COSM Research Grant, which helps support my graduate student Alex's thesis research. And with that, I want to thank you all for coming today. I hope you enjoyed the talk, and I would be happy to take any questions you may have.
Awesome, Heather. Thank you so much. What a neat story. And to me, it's really intriguing to see how different the salt marsh restoration work you're doing just up the coast from us is compared to the work that we're doing here in our part of Florida. So, thank you for sharing a neat tie into the restoration work that we're involved in. And we've already got a whole bunch of cool questions rolling in. So if any of you have questions, go ahead and type them into the Q&A box, and I'm going to go ahead and get started. The first question, does Spartina require insect-driven pollination?
No. So if you look back at the flowers are really undeveloped. And so as with a lot of grasses, Spartina has kind of gone back to relying mostly on wind mechanisms for pollination.
All right. Are there any other hardier grass species that can be introduced that can coexist with Spartina when you do restoration?
So I'm gonna say no, just because our marshes here are predominantly dominated by Spartina. And so I would say yes, if we saw other species mixed into a natural marsh ecosystem, but here we basically see just monoculture stands of Spartina. So introducing another species may actually shift that community structure. We see that once we kind of alleviate some of those abiotic stresses, like soil salinity and low oxygen availability, Spartina is actually really terrible at competing with other plants for resources. So it's kind of their ability to tolerate those conditions that allow them to exist. And so once they have to compete with another plant, they tend to struggle with that.
All right. So one of our viewers noticed that germination rates are low. So they are wondering whether the germination rates that you observed are normal for plants, and is that a typical germination rate, or is the germination rate found in Spartina unusually low compared to other types of plants?
So the germination rate is unusually low. And this is, again, not uncommon in habitats with high abiotic stress and rapid erosion and things like this. It's hard for a seedling to survive. It's actually not uncommon in Spartina to see such low germination rates, but these are extremely low compared to what we see for a lot of other plant species. So we're trying to bring that number up, but they are primarily reproducing through vegetative propagation, even though they have such a high genetic diversity. So that's kind of just a product of working with a special plant species like Spartina.
All right, so another somewhat related question. In nature, do these grasses reproduce primarily by seed, or do they use those the underground runners that you showed in the diagram at the start of the presentation?
Well, I don't know how many studies have actually looked at in situ seed production, just because it's such a hard habitat to get to and to track and things like that. But I can say that we expected them to actually have less sexual reproduction than genetic studies have found. So we do know that they seem to reproduce quite easily through these vegetative propagations, but here in Georgia, we have surprisingly high genetic diversity within our populations, which could only be a product of sexual reproduction. So sexual reproduction seems to contribute more to the population than we initially thought.
Great. So we had a couple of different people ask questions about salinity. You found that plants further away from the ocean produce more seeds. If you collect seeds that are from plants found further from the ocean, can those seedlings survive in saltier water closer to the ocean? And are there any studies underway to try to grow seedlings that are more tolerant to higher salinities?
So it is traditionally, so in traditional nursery production, plants are grown in fresh water. So fresh water is kind of the kind of normal. Most times there has been studies that show that you have to salt harden them before you transplant them into a salt marsh. So you have to kind of build them up to that salt tolerance. We have not done studies about like that yet. We hope to do it soon. Like I said, right now, at least in our wetland, we have an issue of kind of maintaining our salinity. But it could be that these seedlings either are initially grown in these kind of saltier conditions or they're salt hardened. That is a question, whether we can take some of these seedlings from these lower marine influence places and plant them in higher salinity. My gut instinct says that yes, we can harden them to kind of prepare themselves for that, but that is a good question and something that we are interested in looking at.
All right, pretty straightforward question. Did you notice an increase in mosquito populations around your wetland?
No, actually, you know, I think the gnats were the worst at the big boat ramp. So, yeah, and there's not mosquitoes; it's those no-see-ums. You know, honestly, because the breeze, we usually had a breeze right there along those creek banks. The bugs weren't really that bad, except for that Rodney Haw area. That Rodney Haw area was a little horrible. But maybe I was just enjoying being in the mud too much to notice.
All right, we've got some questions here that might be coming from some sciencey people. So can you select for plants that are more resistant to things like pollution, boat wakes, or other problems that your salt marshes are facing?
You know, I don't know. That's a good question. You know, if we could simulate those conditions, we could create a, you know, nursery stock that was more resistant to that. The anthropogenic disturbance is hard to select plants that are prepared for that. But one of the reasons why we want to use local plants is so that they are prepared for those other kind of really kind of regional differences in environmental variables. And so we kind of think that nothing's going to persist better in a Georgia salt marsh than a Georgia plant. I don't know if that answered your question or not.
Absolutely. So we have a question from a postdoc at Northeastern University in Massachusetts, and they noticed that when they went out in the fall of 2021 to collect Spartina seeds for germination, a lot of the seeds were chewed up by insects. She thinks that's plant hoppers doing that, and she's wondering whether you're noticing any herbivory on your seeds in Georgia. And then a different viewer just wonders more generically, are there any grazers on Spartina?
Okay, so there are definitely grazers on Spartina. One of the most well-known is the periwinkle snails, the litorinas. They actually will make little scratches and then farm fungi in the scratches, so they're not actually grazing on the Spartina. But there are plenty of herbivores that graze on the Spartina. And then, like I said, their dead stems are a huge food source for a lot of things like crabs and other detritivores. As for seed herbivory, I haven't really seen that in our marshes. And, you know, not really signs of the seeds themselves that were kind of chewed on. But we did notice in some marshes, we found this kind of like sticky amber substance that was kind of coating these seed spikelets, and we couldn't collect them because they were so sticky. And I have no idea what that was, but that was only found at the one site. Once we got the plants back to the marsh, we also started seeing some herbivory. So we had things eating our plants and eating the roots, and it just became a big fat mess. Yeah, no, I didn't really see seed herbivory in our sites.
Right, do you have any plans to minimize the introduction of pests and other organisms when you inoculate your study containers with sediments, the wild sediments that you're going to try to use in the future?
Yeah, so it's one thing that's hard to control for. Oftentimes when I do these soil transplant studies, I'll sterilize the soil, but that kind of defeats our purpose because we actually want those microbes. So yeah, we may introduce some pathogens within. We will have, each plant will be in a pot, so we will have limited movement, exchange of soil material. But one of the interesting things, I don't know how related this is or a side note, is I've grown plants from other sources. Like I've gotten plants from Tennessee and grown them here in Georgia. And when I grow plants for these Spartina plants from other sources, they get devastated by pathogens. I've had plants covered in aphids. I've had plants succumb to rust and other fungal diseases. But on these native plants that I've been growing, I have seen limited pathogens and limited insect infestations. So it's something kind of really interesting that they may be better suited also because they have greater resistance to these pathogens. They're natural pathogens. So it's an interesting kind of part of it that we're looking to maybe tease apart later.
Great. Just a couple more questions before we wrap up. How deep is the water where Spartina grows?
So our grasses get fairly almost completely inundated during high tide. So we actually have our high tide can change about 8 feet. So during high tide, some of these marsh sites, just the tips of the grass will be sticking out. So they can get pretty much almost completely submerged during high tide.
When you showed salt measurements, what do the whole number measurements equate to? Are they parts per million?
Yeah, so there are several ways that you can it's the same thing. There's been a debate about how to report salinity. And so and it really depends on what journal you're writing for sometimes, right? So sometimes they just want whole numbers, but it does equate to parts per thousand or parts per million. So 35 would be ocean, pure salt water.
Great. So one of our viewers noticed a map. I think it was one of your first slides that had lots of green in places away from the ocean, including Minnesota. And they're wondering whether there are any salt marshes in areas that are distant from salt water, like the Great Lakes, for example.
So there are these. I know that there are a few areas where there are some more inland saline wetlands. But most of those are coastal wetlands are freshwater that they showed that was kind of surrounding the Great Lakes. That map there didn't really distinguish between coastal and freshwater from the USGS. It was looking more at just wetland in general.
All right. Last question is kind of a big picture question. Are marsh die-offs part of a naturally occurring cycle, or is it really human-caused pollution that's driving these die-offs?
That's a good question. So the last die-off event I think that occurred here in Georgia was in the mid 2010s, maybe like 2012 or so. And there was a lot of research to kind of determine what was causing these big die-off events. They thought it might be grazing pressure. And so they actually thought that the periwinkle snails may be having a negative impact and causing these die-offs. But the kind of data has suggested that these die-off events seem to be correlated to droughts. And so when you have these drought conditions during the growing season of Spartina, which usually occurs kind of in our summer, which is more rainy, it can cause changes to the chemistry of the soil. And that kind of drought related changes in the soil chemistry is what is being implicated in these die-off events.
All right, Heather, thank you again. It was such a cool program, and it was really, really neat to see how you guys are doing things just one state north of us. I know our audience appreciates seeing that comparison. For all of my audience members, remember, we only have 2 more lectures left. Next week, we have Dr. Chelsea Bennis to tell us all about the octopuses of South Florida. Incredible underwater footage that she's going to share with us. Definitely going to be a neat program. And then 2 weeks from tonight, again, we have this hybrid lecture where if you're interested, you are able to attend in person at the Coastal Center for the first time this season. Just make sure you go onto our lecture series website and register again if you wish to attend in person. Space is limited. We only have a limited capacity, and we don't want to pack the house too full. But if you sign up soon, you should be able to attend with us. All right, everybody, have a great night. Thank you again for participating. And Heather, thank you for spending your night with us.
Thank you all for coming. I really appreciate it. And I'm happy to answer any other questions. Just shoot me an email. Thank you.